Good morning and welcome to the webinar on the effects of the predatory mite, Amblyseius swirskii, and its byproducts on the whitefly, Bemisia tabaci population. This is the second webinar in a series on biological control of vegetables in the Southeast as part of a NIFA OREI funded project. So for today's webinar, I'm very pleased to welcome back Dr. Oscar Liburd, who is a professor in the Entomology and Nematology Department at the University of Florida. His expertise is in agricultural pest management, and he works with organic vegetable growers, primarily on cucurbits, cole crops, and solanaceous crops. Dr. Liburd also works in the area of small fruit pest management. So welcome back, Dr. Liburd. I'm going to turn off my video and then hand things over to you.
Good morning or good afternoon, depending on where you are. Again, I'm Oscar Liburd. I'm responsible for organic vegetables and also small fruit pest management here at the University of Florida, based in Gainesville. I want to acknowledge, first of all, the contribution of one of my former students, and she's now a postdoc at Virginia Tech, which is Lorena Lopez, because a lot of the work I'm presenting today was done by Lorena Lopez, and also Professor Rosangela Maurice, and she did a 6-month sabbatical in my lab. She's from Brazil, at University in Brazil. Again, this work was done over the past 3 years, and the work was funded by the OREI project, which primarily look at things like the effects of predatory mites and key pests of vegetables, whether it's whiteflies or thrips. One of the first things I would like to do, because a lot of the discussion is focused on Amblyseius swirskii, is to talk a little bit about just a brief background on Amblyseius swirskii. Well, vegetable growers, especially organic growers, tend to use this predatory mite for the purpose of management. And one of the reasons, because they don't have a lot of options. In other words, they can't apply pesticides like the standard conventional grows. So they are looking at, or they're trying to determine the effectiveness of this predatory mite. And it's a relatively new predatory mite that we started using just a couple of years ago in the vegetable system. Before that, we were using some other types of mite predators. And so there's a lot of curiosity to find out exactly what is the effects of this predatory mite and key pests such as whiteflies. Just to give you some background here, this is a beneficial predatory mite that is endemic to the Eastern Mediterranean region. When I say Mediterranean region, I'm talking about places like Turkey, Greece, Cyprus, and Egypt. It is a generalist feeder, and we use it quite a bit to control things like whiteflies and thrips, especially in greenhouse. However, my laboratory, and I think maybe 1 or 2 other laboratories in the Southeast, are looking at the possibility of using this mite in outside in field operations as opposed to just focusing on greenhouse. One of the benefits of using this mite is, as I said before, it's a generalist predator. It means that it can feed on other things besides mites, or other things besides a pest like thrips or whiteflies. It tends to feed on pollen, and we want to take advantage of that because it means then when the pest population is very low, this mite would still be able to survive. It can also be applied what we call preventatively and also curatively. When we say preventatively, we mean that before the problem developed or before the pest problem developed, we could apply this mite. And what happens, it goes into the system, and if there's not sufficient pest, it will feed on things like pollen. And when the pest appears in the system, then the mite would go after it and potentially reduce the population below the threshold level. And curatively, in other words, if you have a high population of the pest, you could also apply this mite. The focus today would be on silverleaf whitefly, although this mite has been used quite a bit on thrips. Just to give you a little bit of background on silverleaf whitefly, it's a very important pest of agricultural crops and also ornamentals. Things like cucurbits, the squash, watermelon, pumpkins, and so forth. They're very susceptible, at least here in the southeast, to silverleaf whitefly. And one of the biggest problems is that the adults tend to secrete sugary substance, and what happens after some period of time, the sooty mole tends to grow on this, what we call honeydew. This sugary substance is called honeydew, and sooty mold tends to grow on top of it, and this interferes with photosynthesis. The plant is not able to carry out photosynthesis, and this can contribute to a significant reduction in terms of yield. One of the biggest problems with the silverleaf whitefly is the fact that it causes a silverleaf disorder, and that's why the name silverleaf whitefly. If you look at the photograph on the left-hand side at the bottom of the page, you see those silvering on the leaf, and this is really caused by the nymphal feeding, or the immature whiteflies feeding on the plant. And what happens after a period of time, you get this silvering appearance. Another biggest problem we have with the silverleaf whitefly, it tends to spread it's a vector for many viruses. In fact, I understand that it can spread up to about 400 viruses. But the biggest problem we have here in the southeastern part of the US is one which is referred to as cucurbit leaf crumble virus. This virus was actually first recorded in Florida in my lab. I had a graduate student back in 2007, and she just accidentally went to the field, and she was sampling, and she saw all these leaves with this crumbling appearance, so she wanted to know what it was. And we sent it out for identification, and they came back saying that it was the first case of cucurbit leaf crumble virus in Florida. So that was interesting. But now you find that it's widespread. I mean, anywhere you go in Florida where they're growing cucurbits, especially squash, you have this cucurbit leaf crumble virus. I think it's the most severe virus affecting the squash group in Florida. One of the things that we wanted to do is to look at the possibility of using Amblyseius swirskii to reduce the whitefly pressure. And we wanted to measure or to find out if this predator could prevent some of this virus transmission. But in order for us to ensure that the predators would be in the field, we decided to use sweet Alyssium, which is the white flower you see there in the background. The white flower there is sweet Alyssium. Sweet Alyssium is an annual plant. It belongs to the Brassica family. It's used mostly as an ornamental, but a lot of growers tend to put it in their field because, again, it attracts a lot of beneficial insects, mostly parasitoids and predators. And we wanted to figure out if we put this into the field, the sweet Alyssium, if it could help reduce the population of the whiteflies. So there you can see one of those squash fields here. You can see sweet Alyssium planted in the center of the field, and we have squash on both sides. And we did not know how much sweet Alyssium to use in order to probably get some kind of suppression. But what we did, I'm showing you just one part of the research. We looked at different size or different population of plants to determine if the sweet alicium does have an effect. And again, the reason why we wanted to do this is because a lot of the organic growers tend to plant it with the intention or with the feeling that this would help them in suppressing some of the whiteflies or other pest problems within the field. So our goal here was to evaluate the movement and performance of Amblyseius swirskii on silverleaf whitefly in field-grown organic squash. Again, a lot of this work is done in the greenhouse, and the field aspect of it is relatively new. So again, this was advice from the growers. They were looking at different varieties of squash, or different cultivars of squash. Cash flow, gold rush, Zephyr, and a tall, and obviously the sweet alicium that we use was the varieties was referred to as tall white. So we look at these three different squash cultivars and tall white variety for the sweet alicium. This is just a pictorial view of how the field looks. I'm going to give you some measurement in the following slide. But you can see that the dark green here represent the cash flow, and then we have Zephyr, one of the cultivars of squash, which is a lighter green. The gold rush was in a lighter yellow, and then the sweet alicium was in a dark yellow. The area where we released the predatory mite is marked in red, and this is the layout on the right-hand side of the field is marked in red, indicating the area where we released the predatory mite, Amblyseius swirskii. Again, we wanted to see how far it can move in the field, and what is the effect of this predatory mite on whitefly population. And in this, there you can see what we did in the field. We georeferenced the area where we released the predatory mites. Again, it's in red. And we also georeferenced some plants, or the squash plants in the field where we were going to be doing all of the sampling and so forth in the field. So just to give you an idea as to where we were able to collect the predatory mites, again, the release area is only in the red. You can see that the furthest point was 121 feet from this is one field here, which is referred to as the treatment field, where the predatory mite was released. And the field where there was no predatory mite release is referred to as the control, because there was no pest management in the control. Again, when we look, in fact, we did not expect the predatory mite to go all the way to the control, because these plots were 27 feet apart. But this was kind of surprising to us that the predatory mite, over a period of 6 weeks, was able to move all the way into the control, and that affected some of the results that we got from the study. So you can see the dimensions here. It not only moved across the field, but it also moved in the north-south direction, the predatory mite. And we're going to be talking a little bit more about that as we go on. Notice where the sweet alicium is in the field. The areas which is in dark yellow, or the dark yellow, is where the sweet alicium is planted. All of the others rows represent cucurbits, cucurbit plots. Now, these graphs gives you an indication as to what was happening in the field as we sample throughout. The first sampling period was on the October 17th, 2018. And you can see in the control where there wasn't any predatory mite release, that the population of whiteflies there was extremely high. The darker the colors, as you can see on the right-hand side, it represents higher population of whitefly eggs in the uppercase, and the lowercase, or at the bottom of the screen, we have predatory mites. So again, at the top you have whitefly eggs per leaf, and at the very bottom we have predatory mites. And you can see the range, the numbers of predatory mites that we saw from For example, where you have the blue, we have 0 to 5 whiteflies per leaf. And again, blue represents 0 to 1 predatory mite per leaf. And where you have the dark red, we have 91 to 100 whitefly eggs per leaf, and vice versa, and so forth. But you can see what is going on here, that early in the season, the control had really high populations of whitefly. And at the same time, if you were to look down at the predatory mites, there were hardly any predatory mites there because the predatory mites wasn't established yet. But as we continue to sample, like the second sampling period, which is 1024, you realize that the whitefly numbers, or the number of whitefly eggs, started to decrease. And when you look at the predatory mites, you realize in the treatment that the predatory mites started to get established and so forth. And if you were to look all the way down to about the third sampling, or the third time we looked in the field, you realize that both the control and the treatment are not showing much. And the reason for this is what I explained earlier, is because some of the predatory mites was so aggressive that made it all the way over in the control. When all the food in the treatment was finished, they start moving into the control because they're looking for whiteflies. So they actually not only wipe out the treatment plots, but it also wipe out the control plots. And this is why I tell you it affected some of the results we got. But the important thing here is that you're able to see that the predatory mites were very effective in reducing the whitefly eggs per leaf. And the predatory mites also became very established. And not only established, but it was able to move into untreated areas and control the whiteflies. This is just a graphical representation as to what happened here. Whiteflies, and again, remember I discussed earlier, we measured things like silverleaf disorder, because silverleaf disorder interfere with photosynthesis, and we wanted to see what was going on there. And we also measure the reduction in terms of the virus. So when we look at the first graph on the left there, you can see the control treatment group. And then you also saw the treatment. Again, the only difference between those two plots is that one had predatory mites, the other had no pest management. The control had no pest management. And you can see the population of, in the darker yellow here, of predatory mites in the control here became high. This is throughout the entire season. But the most important thing is that the whitefly population, whitefly immatures, was also relatively high, and whitefly eggs were also very high in the control. Whereas if you look at the treatment group, you realize there's significant reduction in terms of all three. Obviously, the reason why we have less predatory mites in the treatment here is because what happened as the population of whiteflies dropped. We have the same relationship whereby the population of predatory mites would drop as well, because they're going to be moving out of these plots into the control. When you look at the silverleaf disorder, again, this was significant. The control had a lot more silverleaf disorder than the treatment. It was significant. So we see there that the release of the predatory mites in the treatment was able to reduce silverleaf disorder. Whether or not this is sufficient to significantly reduce photosynthesis, we're not sure about that, but at least it was significant here. One of the things I wanted to discuss here also, this is some data taken while we were doing this experiment. There were some questions that growers raised about how should you release the predatory mites here. And I just wanted to show you this graph whereby whether you release the predatory mites, as you can see on the right-hand side here, the top right-hand side, whether you release the predatory mite on the leaves or you use something they call sachets, it did not make a difference. Or you release it on the stem, it did not make a difference in terms of the number. This is the bottom figure now, the bottom figure on the right, it did not make a difference in terms of the number of whiteflies it was able to reduce. However, in terms of recovery, you saw that we found more Amblyseius swirskii or more predatory mites on the leaves compared to the sachets, stem, and the control. I guess if you were to look at this maybe long term, maybe releasing on the leaves, you will see more reduction in terms of whitefly. But this experiment was done over about six to 8 weeks. But what we saw over 6 to 8 weeks, again, there wasn't any reduction in terms of the whitefly population there. But we recovered more on the leaves and the sachets, so to speak. What are the sachets? You can see on the top right-hand side there, these are, if you want to call it like paper bags, small paper bags, and this paper bag has only a small amount of mites inside there, but it also have food inside there for those mites to feed on. This is completely different to just releasing the mites onto the leaf, as you can see in the second photograph there, because what they're saying, if you want to release over a period of time, it might be better to use the sachets. We have not really found that to be true, because we are getting the same results whether we sprinkle the predatory mites directly on the leaf versus using the sachets. So some of the conclusions that we draw from this work here was that Amblyseius swirskii reduced whitefly population, as you see, with squash intercrop with Swirskii. Predatory mites were able to establish and disperse successfully. You can see that the predatory mites even went as far over as the control. And it appears that there is no difference between direct application to the leaves, I'm talking about of the predatory mites to the leaf, as opposed to using the sachets. There's no difference in terms of whether you apply it to the leaf or the sachets. One thing I should tell you, and I forgot when I was discussing the Silverleaf, that we did not find a lot of the predatory mites moving into the Silverleaf. We only release the predatory mites in the squash. We did not release the predatory mites in the Silverleaf, and we did not find a lot there. That was kind of surprising to us because we thought that the aroma, from the Silverleaf and also the attractiveness of the flowers would harbor predatory mites, but that wasn't the case. We didn't find that to be the case. And that was interesting when we were looking at that effect. Now, another factor that we probably looked at here was that what is the significance of adding another predator? So far, we've been looking strictly at Amblyseius swirskii. Well, the question is, what happens if you add another predator to the system? Would that affect the effectiveness of the biological control? And those are some questions we wanted to answer when we did this experiment. This was done in the laboratory, but we think it has strong applications to the field. And basically what we have here, we have 2 Amblyseius swirskii that we released into these jars. We also released another predator, which is Belfastus catalina, because these are questions that viewers ask. Suppose you use 2 predators, what happens? And these are things that we wanted to address. So, and in the third treatment, we looked at 2 Amblyseius swirskii and 2 Belfastus catalina. To tell you a little bit about the Belfastus catalina, this is also a predator which is used to manage whitefly population. In fact, when we started our series of experiments, we did more work with Belfastus catalina, but there are some restrictions or some limitations to using Belfastus catalina, because once we moved to the field, we realized that, especially for Florida weather, it did not do very well in the field. So that's the reason. I mean, we were able to recover some, and we saw a little bit of reduction. But in terms of the overall performance, we did not think it did very well. And that's the reason why we started looking at Amblyseius swirskii. Some growers have adopted using the Delphastus catiline, but again, we just feel like we can do much better. And they wanted to know what happened if you use both Amblyseius swirskii and Delphastus. And so we did these laboratory experiments to figure that out. And in this bioacid chamber, we have two tubes. One has a young squash plant inside there, a young squash plant. And then we released 6 reproductive active whiteflies in the jar, and then we leave them just for 24 hours, and then we remove everything. We remove the whiteflies after 24 hours and see exactly what happens. And basically, what we found was that this is the control where we did not use any predators, obviously, in that treatment group. Here is where we use the second graph represents where we use Amblyseius swirskii, and then we use Delphastus, which is the third or the second predator, and then we use a combination of Amblyseius and Delphastus. Now, this was done in the laboratory, so you have to be very careful in interpreting these results. The Delphastus tend to perform fairly well in the laboratory or in greenhouse operation. In the field, it doesn't perform as well. So that's why I say you have to be very careful in interpreting these results. So we saw numerical a little bit of reduction. It was not significant between Amblyseius swirskii and the control. You can see that there are A's and A's, so it wasn't significant. But Delphastus was able to reduce whitefly pressure significantly. Interestingly, when we add Amblyseius with Delphastus, we saw a little further reduction. Whether this was the performance of just the Delphastus, we're not sure, but we saw further reduction using Amblyseius swirskii and Delphastus. Yeah. So the findings here is that the female whiteflies show different oviposition capacities due to the presence or the absence of the predator species. So again, what we saw was that there were differences in terms of the number of whiteflies with Amblyseius swirskii not performing as well as we thought it would do compared to Delphastus. We were wondering what would happen if we did the same experiment in the field with Delphastus, but we think the results may be different, and we are not sure until we completed this experiment. This was something that we recently did. Also, the number of eggs laid in the presence of Delphastus catiline and Amblyseius swirskii, Delphastus catiline, and a combination of Amblyseius swirskii and Delphastus was fewer compared to the control. So again, you can see some form of reduction going on there. But one thing we noticed while we were doing this experiment was that it seems like that the predators or whiteflies, on the other hand, were sort of not only focusing on the adult predator, but actually focusing on other things in the field. As soon as they can somehow smell the predator, the whiteflies were not performing. It seems like there was a reduction in terms of the number of whiteflies. Oh, they avoid laying their eggs on squash, put it that way, if they can smell the predator. So what we did a small experiment looking at where we had, initially we had 4 Amblyseius swirskii, we had 4 Delphastus catiline, and then we had just 2 predators with Amblyseius swirskii and 2 predators with Delphastus catiline. And then we just actually had an untreated control without any kind of predators. Again, before we actually start the experiment, right? The reason why we have 4 and -2 is because we started the experiment with 4. And then after 24 hours, we took 2 out, so we left with 2. The same thing for swirskii, the same thing with catiline. So we had 2 swirskii, 2 catiline, 2 swirskii and 2 catilinae after 24 hours because we removed the first 2. And one thing we should also tell you that is the fact that where we had 4, we left the wrapping or the paper wrapping. It's actually filter paper inside of the experimental jar or the experimental arena. Where we had the 4 Delphastus catilina, we also left the wrapping material in the experimental jar. Removed 2, but we left the experimental material in the jar. The treatment with 2 predators A. swirskii, they did not have any wrapping, any filter paper or wrapping from the predator. And where we had Delphastus catilina, it did not have any wrapping. So what we found, and just look at the results here. Again, on the x-axis, it says Amblyseius swirskii exposed. Exposed meaning that it had the wrapping. Amblyseius swirskii without the wrapping, when you see just Amblyseius. Then we have Delphastus catilina with the wrapping, it means Delphastus exposed. And we have Delphastus by itself. And what we noticed for some reason, and on the y-axis, we have whitefly eggs, the number of eggs produced by these whiteflies. Again, what we noticed, this was pretty interesting, that although we had the same amount of insects there, the fact that we had the wrapping with when we had 4 in there initially, we removed 2, but we left the wrapping. The fact that we had the wrapping there, it caused a reduction in the number of whitefly eggs. The reason for this is not clear. We're thinking that whiteflies are able to smell the odor from these wrappings, and they're afraid to put their eggs in these areas. But this was interesting for us. And one of the things that when we shared this with one of the suppliers, they were telling us, this is a significant finding, because what you can do also somehow, this is maybe in the future, but right now we're thinking in terms of pest management, is that sometimes some of this paper wrapping that the predator mites comes in, you can probably just put that in the field. Along with the actual predator, and you might be able to see some reduction in terms, depending on your field, you might be able to see some reduction in terms of the number of eggs laid by whiteflies. But this is all new for us. We never saw this before. This is again, we ran into this by accident, and we did not know. We did not expect these kinds of results. Again, the wrapping, the presence of the wrapping caused significant reduction in the number of whitefly eggs. Again, this is pretty much what I'm saying here. When we had Delphastus catilina by itself and Delphastus exposed, we had a reduction in the number of eggs. However, when we have Amblyseius swirskii by itself, there was no reduction. However, when we had Amblyseius swirskii plus the wrapping, we saw a reduction in the number of tabaci, or the number of whitefly eggs. So some of the conclusion we drew from this was that somehow whiteflies are afraid, even though they don't see the predator, they're afraid to lay their eggs in the presence of the odor from these predators, and mostly the Amblyseius swirskii, and in this case, the Delphastus catilina. They're refusing to lay their eggs in the presence. And maybe in long term, some of these byproducts can be used in the area of pest management. I wanted to just, towards the end here of my presentation, I wanted to talk a little bit about these predatory mites, because the argument some people have used is that it's relatively expensive, but in the case of organic growers, they have very limited choice, because they can use a pesticide, but they can only use a pesticide if they have exhausted all other options, all of the cultural control, preventative control, biological control. Then you have some justification to use maybe a pesticide that's labeled for organic use. The problem is that only few pesticides are labeled for organic use, especially for whiteflies. The best one that, based on the research that we have done, the best one that offers some very good potential for controlling whiteflies is MP. less potassium salts. NP has worked very well. The problem is you can only use NP after we have exhausted all of these different options for controlling whiteflies. So you can actually, these are some suppliers where you can get this Amblyseius swirskii, eOrganics, corporate biological systems, and also tip-top biocontrol. All of these companies do have Amblyseius swirskii for whitefly control. So far, based on all of the predatory mites that we have looked at, it appears as if Amblyseius swirskii offer the best potential. You can buy these in bottles of 25,000 or 50,000. You can also buy the sachets for them. These sachets, 50,000 has 200 mini sachets with hooks in them. But each one of these sachets has about 250 mites with them. So this is an option for control. And that's pretty much my last slide I have. I'm going to open the floor for questions. I don't know if Alice has any comments or concerns or questions. This is pretty much all I have for the presentation today.
Okay, yeah, I was just going to share the website of this project, and we have 2 more webinars about this topic coming up in the coming weeks from different institutions and their experiments related to this project, which was called Biocontrol Tactics for Key Vegetable Insect Pests in the Southeastern U.S. It's led by J.C. Chong of Clemson University, and he gave a webinar a couple of weeks ago, which has been recorded on our YouTube channel, which is kind of a general introduction to biocontrol and how you can evaluate which products might be effective to use. So it's kind of a complicated topic, and there's not complete information about it, as JC discussed. So work like what Dr. Liburd is doing in Florida is really important. And so if you have any questions for Dr. Liburd, put any questions into the Q&A box. And while we're waiting for people to do that, I just have a quick question. If you could clarify a little bit more about the relationship between the alyssum and the mites, whether or not, how much effectiveness you would see if you didn't actually plant the alyssum as well?
Well, based on the research that we have done, it indicates that Elysium didn't make much difference.
Right, that's what I heard from the results there.
And that was kind of shocking to us because all of the growers, organic growers, at least here in Florida, they tend to plant sweet Elysium because they think that the sweet Elysium tend to harbor these beneficial insects, not only pollinators but also the predators and the parasitoids and so forth, which I think it does. But in this case, we did not see any movement. In fact, the very few predatory mites we found in the Elysium. We did not want to release any predatory mites in the Elysium because these things are relatively expensive. And we thought the attractiveness of the Elysium would harbor these predatory mites, but that wasn't the case. It would be interesting to see if you release in the sweet Elysium and then you release on the crop, what difference that would make, or just release in the sweet Elysium and see if they would move onto the crop. Those are things that we haven't done yet. All of these things that we are doing is relatively new. So
yeah, that's what I was thinking. And I was also wondering, just sort of year after year, have you seen an increase in the predatory mite population, or do you have to just reapply everything every year?
Well, we do, for the experiment, we do reapply every year, but we can find, when we do our pre-sample, we'll find more predatory mites almost year after year. So somehow they're surviving on other things. Maybe those wildflowers in the field or something, some kind of ornamental plants or something, because normally the field, these organic fields that we use, we would leave it fallow for almost a year before we go back for the second season or something like that. Yeah.
Okay, I don't see any additional questions, but thank you so much, Dr. Liburd, for sharing this presentation with us and for taking the time to be here with us today. So thanks very much.
All right, thank you.